turn white like this if
And
it's,
yeah,
if it's real.
right.
Roger. Okay.
So now we're starting to climb a little bit. We started out at 571 meters, and we're now at
Reach nav, 20 meters,
564.
210.
When we reach the top, we will be at 499 meters, approximately, at our next waypoint. Okay, she just wrote in and said that she saw a Pacific
Yeah, sure.
What are the big starfish that we're seeing here, Meredith?
You want to stop and look?
They look like
Big pink
Pycnopodia.
guys. Pycnogonia?
Pycnopodia. They usually have about 24 legs, large center. They're kind of the Roombas of the sea.
Huh.
Do we want to stop and look at one?
They were pretty decimated by the sea star wasting syndrome.
Yeah.
Along the west coast for some
Yeah,
years.
that's what I thought. So we're seeing
I'm
a lot more.
happy to see them.
And are these sea stars ahead of us as well?
Looks like two of them, yeah.
The white one?
That's a classic Pycnopodia forward of that.
Pycnogonia?
It's like got a P in there.
You want to zoom in there, Ed?
Pycnopodia. Yeah, general rule of thumb, never ask anybody from Philadelphia how to pronounce anything.
Okay.
Not.
Yeah, we really have had a horrible sea star wasting situation going on here on the west coast. It's there's several rumors about what's going on, from warming oceans to viruses, but in deeper water, we're seeing a lot of the species that have now vanished from the shorelines all the way from Alaska down to California.
Can we get a peek at the next one as well, Bob?
Yep.
A suggestion that this might actually be Rathbanaster, not
Pycnopodia.
Pycnopodia.
It's got a lot of spiky type things, which you don't
Thanks,
see.
Meredith.
Yep, thanks, Meredith.
Rathbanaster is one of my favorites.
And then
I
this.
think that I could be wrong about this. The Pycnopodias are called the sunflower.
That's what
They
this.
usually have more pigmentation, I think.
A little
Yeah.
off the top.
Look up.
Boy,
yeah, you can zoom in.
how many
Copy,
legs
going in.
are on that thing? Oh my God.
Okay, everybody at home, count the legs.
Yeah.
No wagering.
9, 10, 11, 12, 13. At least 20. And that's also called the California Sunstar.
Is that all you
That's
got
it.
on the zoom?
Thank you. Oh no. I got stuff like that. I love Mr. Shrimp.
Little shrimp.
Yeah.
Yeah, that's good detail.
Yeah, Rathbanaster are found down to 1,000 meters, and from Alaska to California, you can see it moving pretty quickly there.
Reach nav, 50 meters,
Using its
210.
Its own little hydraulic system, push against it.
All right, moving on.
That's right. That was pretty. Now let's go find some chemosynthetic fauna. Stuff that lives off all the microbes produced by this methane. So we will be seeing chemosynthetic communities around these bubble stream sites.
What's this? What's this right here?
Yeah, these are exciting dives. We're really seeing the whole driver of the system. So we're going to be doing some chemical samples to understand exactly, what the chemical composition of the bubbles coming out, but the transformation of that methane, the chemical reactions that happen there and the microbial processes can turn that into rock. So we get to see kind of the geologic setting, and those transformations make it bioavailable, make it accessible to these other animals and create these food chains. So we're going to see the biological community formed by or formed because of the chemistry, and the geological setting influencing the chemistry. And we're going to check it all out.
Right ahead of us, we see, I'm sure that's a thorny head, crawling around the bottom off to the port side a little bit. I've been informed by my fisheries friends that rockfish do not live deeper than 500 meters. So generally when we see something that looks, rockfish look a lot like a thorny head. So that little guy is probably a thorny head. They tend to kind of crawl along the bottom. And you notice there's just some tiny little outcrop
Wanna
there. You'll
zoom in
find
real
them.
quick there, Ed?
Yeah, going in.
You'll find them next to any kind of structure. I think they think they're hiding.
Yeah,
Very stealth.
I would have called that a rockfish if I didn't know better. The hard thing with rockfish
It's
is
a
Okay,
thorny
we gotta
head.
get moving.
Yep.
You will be corrected.
Yeah, that's very helpful knowing the depth limit because
It's a long spine thorny head. Thank
Rock-
you.
rockfish are so
Meredith.
hard to get correct. There's so
Oh,
many
yeah.
different kinds.
I just like to call them rockfish.
Yeah, that's
Well, there's the yellow eye and the canary, and I used to know them all. We did a lot of work on Hecata Bank with the fisheries group at Hatfield.
Oh, that was octopi.
Had a cheat sheet of it. There's just
so
dozens,
so many.
dozens of them out here, dozens of different types. And perhaps my friend Waldo
more
Wakefield
critters
can
coming
write
up,
in.
a little
Mm-hmm.
rocky debris.
Yep, we're starting to see some rocks, carbonate looking. Okay. So this, what we believe is a carbonate platform, has a sediment layer on top. You can see everything raining down from the sea
I
surface.
upped the gain on the Z
Yeah,
because we're
going.
diving in like it wasn't keeping up with the slope.
Okay.
So we're heading upslope now. We are now at 559 meters. Okay. Okay. Where is that?
Cool, look at all this up here.
Okay.
It's getting steeper here.
Okay, we're
Rockier.
moving up. Lower left quadrant. Did you see this?
Bridge nav, 50 meters, 210.
Yeah, you can see it here. Go down.
Okay.
Yeah, Tim's looking into that sea log question, not logging coming in.
I don't see how to use it. Okay, we're heading up slope. Seeing more and more
Yeah.
rocky features.
So the carbonate ridge overall is maybe 250 meters wide, something like that. Does that seem about right? Maybe 500 at its widest point. It's a large raised feature compared to the seafloor around it, and we are flying up the north. We're sort of on the northeast corner of the northern edge of it, and so we're going to be moving southwest to
Southeast.
southeast to come up.
We're at the northwest. Well, we will be at the northwest point of this ridge when we get to the top, and we will head down to the southeast. It's about 4 kilometers long, but we will not be covering the entire ridge. Quite a bit, about 3/4 of it. And we're expecting to find these bubble stream seep sites
Yeah,
once
it
we
usually
get to
has
the
that
top.
0.5 steps.
And along the way, we get to check out all this life.
Yeah.
The red
I
fish, our longfin thorny
Yeah.
fins.
Yeah, that's kind of like, that's sort of like an easy number to deal with.
Yeah.
You know? It's like not too much.
So if the slope is going up, how high do you want to kind of stay above it?
Well, you want to get a good view,
Okay.
right?
Yeah.
So you can be closer to the bottom if it's flatter.
Yep.
And then once you start, if you're going up slope and you're kind of clipping along, then you kind of want a little bit more buffer, and
See a coral.
Sure.
you can still see the bottom good.
We're seeing a coral ahead of
Yeah.
us.
So you're in auto altitude and auto heading, but
Yeah.
you're driving.
I'm driving, yeah.
Do you know
Not
what
sure
kind of
what type of coral that is, Meredith. You got an idea?
And how
Did you
fast
wanna
is the
look
ship
at it?
moving?
Are we looking at it?
Yeah, let's look at it.
Okay, you
First
wanna
coral we've seen.
Zoom in there, Ed.
Coming in. Holding.
Oh, beautiful.
Some type of
The
bamboo.
bamboo.
Yeah.
Yeah,
Yeah.
it looks like a bamboo coral, and they can
Yeah.
live for a couple of hundred
banded.
years, I believe.
How did everyone make that ID?
Oh, this is an easy one.
It
Looking
looks
at
like bamboo.
the spine or the base of the coral, you can see those black little notches. Almost looks like a bamboo appearance, a bamboo shoot. Isidella tentaculum.
Another coral in the back there, the red guy.
And I don't see an associate. Oh, no, I don't see anybody on this
Okay,
one.
moving on.
Okay.
Oh, why?
So you can hang back until you're like under me and down cam and then move on.
Yeah,
How fast is the ship moving?
like 0.3 or something.
Right now, the move just ended, so.
Okay.
But we've been doing about 0.5.
Okay.
You can see there's some slabby carbonate slabs here. We're seeing more and more debris on the seafloor.
Swimming crinoid, I
Okay,
think.
so the more black it is, the more the slope is?
Less sediment.
Sometimes you get a bright like
sediment.
that.
Yeah.
That's the rock in front
Pull
of you,
an
and then there's
enemy.
a shadow behind
Oh.
it.
Yeah.
Raj.
Yep, there's a sea anemone. You can notice that on these carbonate slabs, rocks, we're seeing a lot more animals than we saw on just the sedimented seafloor.
Okay, coming up. Oh,
Yeah.
Roger.
steep. Getting
Bridge nav,
steeper.
50 meters, 210.
What are these little guys that we're seeing?
A lot more corals.
Yeah,
now.
we're seeing little branches of coral,
Okay. Yep, a lot more corals.
brittle stars.
Thorny heads.
Oh, big an enemy. Hi. Cool.
Wow, that's
Urchin.
beautiful. Oh, maybe we should.
Wow.
That almost looks basaltic. It's so black. But basalt isn't slabby like that.
So a lot of hard substrate specialists, meaning animals that need something hard to settle onto, like a
Right.
coral or an anemone. So as we change from that soft, deep sediment where you have sediment specialists to a hard bottom relief where you have hard bottom specialists, you see a change in the community and a big bump in at least the macrofaunal diversity. So there are all kinds of tiny things that live in the sediment that we're not seeing as we fly
Right.
over, but as far as big creatures, really a step up here.
Yeah, it's still full wide. If I'm, like normally.
The starfish that we're seeing are Rathbanaster,
No, that's got
is
it
their
up
genus
down.
name.
Like try and bring it down. Sometimes you could go.
I think we, Bob, when we see something, one of these slabs that we could pick up,
Green immediately
we might want to do a grab,
to try and add
Okay.
some reds in.
You wanna-
Yeah.
take our first sample, and maybe
are
we
you
get a better
particular
idea.
about the spot, or
I'm not picky about the spot, just whatever's
can
easy
we-
for you
how
to
far
sample.
move we got?
Looks like we have about 40 meters left.
Okay, I'm gonna zip ahead a little bit and
Sure,
see what's happening.
I'm sure there's more
What
ahead.
size do you need?
Not that large.
Okay,
We don't want to weigh you down. These rocks are bigger
want a
than they
flat
look.
one. You want a flat one.
They are.
No lumpy bits. You want flat.
I get down there, it's as big as the milk crate.
Yeah, exactly.
And where's that gonna go? Starboard side?
So yeah, we have the starboard box and the forward. So whatever.
Well,
It's easy,
are we
it's-
collecting clams later? Those are better up forward. So
We probably will take a grab, yeah.
Yeah, so we could
So-
do the starboard.
Yeah, and like I said, it doesn't have to be big because we'd like to get a couple of samples.
okay.
We'll take one here, and then we'll probably take one towards the
We'll
end
look
of the
for
dive.
something flat coming up here.
Are they the ones
These are
with,
probably broken off of carbonate plates.
So you wanna see a big slab with breakable things?
You mean with other animals on it?
Well, no. You want something that's not falling off, or you're okay with a piece?
It would be better if it's in place, but I think this all comes from the same source.
It's all way up slope still.
Right.
Okay. So any old rock that's flat?
Any old rock.
Okay.
Hey, let's try the draw. See if I can do it. Now let's get something little, something over, like something over here.
Bridge nav, hold position.
Okay. That works.
So we're gonna go in and collect one of these pieces of rock that we're seeing strewn all over this slope.
You're looking for something up in this area?
Sure.
Okay.
I mean, it'd be best if it's attached, but if not,
Well, so you're not okay with these, or
I'm okay with those, Bob.
okay?
Yeah, they're looking up in
Kylie's
this
gonna
area,
do
Bob,
the grab.
up there.
Looking what?
They're looking at these over here.
Those over there. Okay.
Can
Okay,
you zoom
hang
in a
on,
little
Kylie.
bit?
Let me scoot over a little bit.
okay.
So you got to be careful you don't hit the triggers on these bottles. That's a bad thing.
Okay.
Something in there.
Sure. If you see anything in place, that'd be good. If not,
You want to point at it?
okay. Hard to choose. There's so many of them.
I can give you
Uh,
a zoom if that
Do you want to
helps.
stop the ship move?
This ship is stopped.
Okay, thanks. Okay.
Want to push there, Bob? Wanna zoom?
Or maybe here,
Yeah,
anywhere.
you You want to push in, Kylie? You're on the,
Okay, Raj.
you got to tell them what to do.
Okay, come in closer, please.
Coming in, holding, focusing.
Yeah, that right there, that looks good. Pretty big.
Come out a little bit so you can see the whole thing, Kylie.
Yeah, that looks
How do
like it's
I
in
Lock
place.
my draw.
Cool.
Yeah, lock on the far side.
Yeah,
And
just
look, always
break.
look at your console, make sure it's locked.
Grip lock,
So we're
Raj.
Okay.
breaking off a chunk. This rock seems to be in place. You can break it up a little more if you want to, Bob, if it's too big.
Come wide video.
Coming out with you.
And it's going in the
Yes.
starboard.
You're going starboard.
Rotate a little bit and then look
Can
and
we
see
zoom
what
in
you
on
got.
it, Bob?
Yeah.
Before we put it in the
Yes.
basket?
Okay, video zoom.
Yeah, coming in.
Let's do a zoom and take a let's pick,
And then
Turn
you
it.
rotate it slowly.
rotate it, yeah.
Holding.
It's got a yellow staining on it. It's probably the size of,
You want the lasers?
Yeah, like
sure, yeah, let's get some lasers on it. Okay, so it looks like it's about 10 centimeters across, right? A little bit more,
Yeah.
maybe
Yeah.
10 centimeters across, maybe 15 centimeters long.
Yeah.
Some yellow staining. Looks good.
All right.
Okay.
Okay, coming out.
Okay.
Full wide.
Okay.
So we'll put it in the starboard bio box.
Yeah. So
And-
you gotta watch out when you come around that. First, you don't want to bump into the light bar up here,
Okay.
so you're clear of it.
Okay.
Okay. So you can come up, so you don't run into the slope.
Okay.
So come up on the shoulder,
Okay.
and then you're just gonna make a smooth rotate
Swing.
around.
And you'll
right?
follow me with the bubble a little bit?
Yep.
Okay. Okay, so shoulder up a little bit. I think I'm full shoulder up. Not that.
That's. You can bring the elbow up a little bit.
Okay. Like that?
So now we're
Good?
Yep. And then start swinging slowly.
Okay.
placing a sample in what we would call a bio box, but this is not biology.
The geo
What do you
box.
call it here?
yeah, it's called the starboard
Can you
bio
go to
box.
sample mode
Okay.
and I'll
It's
look at
called
the bubble?
starboard box.
Okay. Switching the sample.
Rock box that also works.
And let me, cameras, we're going, starboard bio box.
And our depth
Yeah,
here is
looking good.
539 meters.
Keep going,
What the box at?
right?
I lift it.
You ready for drawer?
It might have lifted up high enough in my wrist.
Well, yeah, you gotta just come up on the elbows some, and so now you gotta straighten your wrist out so that you're on the other side. Yeah, there you go,
Okay.
like that. And then
Okay.
come up a little bit,
Yeah, I'm ready for
Okay,
bio box out.
and then I'm gonna go out on the bio box.
So we're a little bit
We're
more
gonna
than
put it in the back somewhere.
Okay.
halfway up the slope here. We're taking a sample of all of this scattered.
One of those, I think
Rocks.
if we're going one of the small boxes.
Okay.
That we believe are carbonates.
So
We tried to get one that was in place.
Yeah, maybe rotate the jaw around a little bit. Yeah, you can see better.
okay, so how do I, when I'm ready, how do I just hit the left?
Yeah, if you just
Okay.
release you should like squeeze the trigger and then hit the button, and then you can gradually let off the trigger.
Okay. Squeeze the trigger, then hit the button when
Yeah.
I'm ready.
Yep.
So just line it up first.
It's gonna crumble, I
gonna-
bet.
Well, you can once you got
See
the
that piece of
once
it
you got
just
it
fell off.
squeezed, then you can release the grip lock, and now you have a hold of it still, right?
Right.
And then when you release the trigger, it'll drop.
Okay.
But
yeah.
you got to keep it squeezed before
Okay.
you let off the grip lock.
You
And I'm
might want
putting
to
it
try
in
and put
D.
it in
You want to go in, yeah.
B because you
B.
dropped some of it in there.
Okay, Roger.
So try
You
to
might
go
want to
for-
get a little
for B.
closer.
Yeah.
It's gonna break
You
up.
got to
Okay.
get down there on it.
And that'll be sample 001.
We're always training someone out here. Today I'm in training.
You're doing
Taylor
a great
Ann
job.
and Megan are helping me out. And it's not as easy as it looks
Nope.
to manipulate these things.
You get to rotate it around so it's flat, and then set it on top, and then you can just kind of nudge it in. But that's good right there.
And then trigger
So
Right.
squeeze the trigger,
Yeah.
and then hit the grip release.
grip button.
Yeah, and then start letting
Oh
the
no,
trigger.
Kinda
I
sliding.
lost my position.
Okay, you still got a grip on
Yeah, I do. So grip lock, right?
it? You locked on there?
Yeah,
Yep.
I am.
Okay. So rotate it around so it's,
That way.
yeah.
Okay.
Maybe come all the way around so that it's coming out. Yeah, but come up a little. And then, if you can get the nose of the thing in the,
Oh, Roger.
then it'll for sure go in there.
How do I get the nose in it? Okay, Bob, I think I'm going to pass it to you.
Yeah, you got to come down on the shoulder here.
Down on
There you go.
Oh,
Hey.
okay.
Just the way you planned
Oh,
it.
yep. Okay, box in, please.
You weren't using the shoulder.
No, I wasn't. I don't know. Okay,
Okay.
yeah, I forgot that was a
So
joint
bring
I had.
bring it up and out without hitting the tube cores.
So Ed,
Yes.
That was pretty good.
I got a request for when we're doing these transits to have the lasers on.
Roger
Okay.
that.
Yeah.
That's a pilot thing, but we'll absolutely do that.
It helps people that are examining the video later on to
Yeah.
get an idea.
Sure.
So if it's all right, that would be great. And
So yeah, just
Swing,
swing it around.
Roger.
Coming.
I know I had to reset.
So
And you want me to just stow it?
okay, I'm coming back to dive mode.
Please, yeah.
good job. Thank you.
You are welcome, Susan. That was my first sample.
All
Hey,
right.
congrats. That was great.
Lots of firsts.
No, she didn't tell us
Can't
that
see it.
before you did it.
Right.
Bob, can you put the, do you want me to just lock it here?
Yeah.
Or
you want me
what-
to stow it?
Well, yeah, I can't see
Or
it.
just bump the camera over to the right.
Yeah. Can
Well,
move-
you need to.
Can you move the pan, tilt, and zoom, please?
Well, it won't go down. It won't go over there anymore.
How about the Herc?
Oh.
Or the Zeus?
That's what I want. That's what I want.
It won't go further over that way. That's what I'm telling
Oh,
you.
it's all the way over there?
Yeah, it's as far as it goes.
Raj, here, you
Just
park
kill it.
You
Hit
got
the
it.
button.
Gee, what?
Here.
All right, we're already a dysfunctional family, and it's the first dive.
That was pretty good for first sample, though,
You did
Yeah.
great.
Why is it moving?
because they killed it.
Oh.
You killed
Oh,
it.
Roger.
But that's okay, because it puts it basically where you want it.
Okay. Okay.
Nice.
Oh, thank you. So we've turned the lasers on, which will help give us an idea of the size of what we're looking at on the seafloor.
Oh my God.
Whoa,
Whoop.
whoa.
Oop, hey, that's one of the
Uh-oh.
seafloor markers I was talking about. We don't want that here.
No. My bad. That
Oh,
was-
Bob.
I bumped the freeze button.
Ooh,
Now that's going to be in our face.
no.
Well, let's stick it in the box, maybe.
Yeah.
It's
You
gonna
want
be
to rack
annoying.
back?
Yeah.
Yeah, Mongo hold it.
Yeah. Maybe.
That's one of the seafloor markers that we hope to put in a spot where we actually find bubble streams on the seafloor.
That's a very nice marker. That's
Not
the nicest
here.
marker I've ever seen.
Well,
Pretty
thank
fancy.
you.
Yeah.
Made by the engineers at Pacific Marine Environmental Lab in Seattle.
Want
Nice.
the tool tray out? Are you
Well, that's not good.
Yeah, we'll tool tray out.
okay? Coming out.
Ah. Yeah.
I have faith in you, Bob. We've been through
Glad
worse.
somebody does.
You want tool tray out more?
Yeah.
Coming out.
I don't know if I can make this happen, but we'll try it.
I can do tool tray in slow.
Yeah.
Slowly coming in.
Let me tilt down a little bit. Hang on.
Oh, there you go.
Okay.
Coming in.
Yep. They keep coming in?
It's stuck on your
wait.
fingers.
Oh, dang it. That's too fancy.
Oh,
man.
no.
Oh. Geez.
Hey, it's number
Coming in.
262. All right, look at that.
Well, we're not in yet.
No!
Oh! Wait. Okay, go. Let me.
They float.
Okay.
That's the point. Thank you. Good job.
So, yeah. So we're not opening that box till we're ready to deploy that marker.
That's right. Definitely. And that'll be the first one we deploy. Let's get up there and see those bubble streams. Ah, the joys. So we have, let's see, about 40 more meters till we get to the top of this
All right,
first little
moving on.
mound. And then we can look around a little bit and then proceed to the area where we saw
Rage nav,
big
50
bubble
meters
streams.
to 10.
When they hit dive, hold on.
So right now we do have the lasers on. You can see the two
Oh,
little
can you
tiny
put bubble cam back on the.
green dots. They're 10 centimeters apart. So they give us some kind of idea of size on the seafloor. So it looks like we got some kind of a maybe barrel sponge up ahead. I'm not sure, but lots of scattered, I would say, carbonate debris on the seafloor. Yeah. Can we zoom in on this
Yep.
sponge ahead?
Ed, can you zoom in?
Coming in.
I believe that that's a barrel sponge. Certainly looks like it. Oh my, it's coming back to me now. Thank you. Beautiful. Thank you.
That all you need?
Sure.
Okay.
Coming
and
out.
The other little guys around him?
Brittle stars.
Those are brittle stars. Okay. That were surrounding the
Full wide.
barrel sponge there.
Push back in. Yes.
Looks like something that would be really pretty with for a vase with flowers in it. We're continuing upslope. Seen a lot of brittle stars, other types of starfish.
Crinoid. Yeah,
You can talk.
yeah,
please.
the crinoids are the ones that are a little fuzzy, I believe. Yeah. Seen some. Oh, look, there's another sponge.
So Taylor Ann, if you want to speak, please do. I'm having a hard time hearing you. You might want to turn it up.
Move a little closer.
That's better, yeah. Continuing up slope, seeing quite a bit of sediment.
Hey,
Lots
Kylie.
of,
I know you've already fired it, but can you hit the dive salvo again as a test for me, please?
okay, some kind of a fish
Bob,
there.
I'm going to switch to dive salvo.
It shouldn't change anything up here. You shouldn't notice it.
Yeah, I just hit it
Okay.
a second ago.
Yeah, I've just edited it. Thanks so
So
much.
brittle stars, here we see its thorny head
Yeah, we're good. Thank you.
hiding behind a rock.
Hold on.
That's a bad idea.
We're seeing some what appear to be carbonate plates.
Bridge nav, 50 meters, 210.
Scattered little thorny heads, echinoderms, starfish. Oh, here we go. In front of us, we're seeing, I believe that's a sablefish. We have a group at Hatfield Marine Science Center that studies sablefish, which is where I work, and Tamara Baumberger and Jeff Beeson were also on this cruise. So yeah, we're seeing several sablefish. Continuing up slope. Still seeing scattered what looks to be some corals. Another one of those barrel sponges. Starfish. Thorny heads. Lots of slabs. Okay, now we're coming up even more. Another beautiful coral just ahead. Maybe a zoom on that and the sablefish.
Zoom in.
Coming in.
And that does not look like a bamboo coral, does it?
No.
No. Thank you.
Coming out.
Moving on.
Moving on.
Full wide.
Now we're seeing bigger and bigger, almost boulder size slabs. About 25 meters from the top of this ridge, which we believe is carbonate. From the data that we collected last night, we can look at the backscatter, the intensity of the returns, and if it's hard rock, you get a higher intensity return, and this came back as hard rock. The only other thing that it could possibly be is basalt, which is oceanic crust, and that is very rare out on the accretionary margin. We do have some basaltic rock along the Oregon coast. It's remnants of old volcanoes, but not a lot of basaltic outcrops here. Generally, you might find a mud volcano, which is a little bit not like what you would think of on land, but comes from a deeper source, deeper in the crust. So it can have a mixed geology, but here we're thinking most of these carbonates are caused from the methanogens, the methane microbial creatures. Kind of eating in the soil and hanging around for a long time and creating these amazing outcrops. That's some kind of an odd sponge. Can't hear you.
Oh, I think it was an anemone. Just
Anemone?
retracted.
Reach nav, 50 meters, 210.
Right there. I think that's an anemone.
Or a mushroom.
Maybe a mushroom coral. Okay. Wow. One of those big sea stars just lazing on a rock. Now that's an anemone. That was a rathbanaster. Okay, so now you can actually see these slabs, which is sort of indicative of carbonates. And here's kind of a more heavily sedimented area. You notice everything that's falling from the sea surface. That's
Video zoom.
what sediments this
Video zoom.
seafloor.
Zoom?
Yeah, please.
Please. Sablefish just swam by.
Oops.
Oh my gosh, there's a hagfish off down to the bottom. And can anybody identify, is that an anemone?
I believe it is a mushroom coral. If you can see here, I have pictures right here.
And
so yeah,
there,
that is a mushroom.
Video wide.
you notice the thing crawling on the seafloor, that's a hagfish, which is, I hate to say it, quite disgusting. We've collected them on previous cruises, and when you pull them out of the bio box, they just exude this sea snot. The only way I can put it. Some people like to eat them, I guess. Thank you for that zoom. That had a lot of biota. So I believe we're looking at carbonate slabs, but, getting larger. More sediment interspersed. It would be interesting to poke that and see how deep that sediment is.
Sure.
We have a stick of poking.
You have a poking stick?
Yeah, we got a pokey stick.
Let's see how
How
deep.
do you?
How wide?
You just need to land.
I mean,
and
we're
get it
thinking
How do
there's.
you land without getting
Reach
it
nav,
all
hold
everywhere?
position.
We're just
Yeah,
gonna
you can just
stop
look at
for a minute.
That and just
Yeah.
come down easy.
Try to get an idea of how thick that sediment is. The multi-beam data
Okay,
that we
now give it a little bit of down
Yeah.
and then hit stick lock.
collect can see through the sediments, but not very far as the backscatter goes. We think there's just a thin layer of sediments here. We'll see how far the pokey stick goes down.
Do you want the
It'll give us an idea of
light
how
on? I
deep.
think it might be off.
I see the pokey stick very easily, huh?
You saw this.
Maybe, here, push the camera back out.
Yeah, sure.
If I can. Yeah, if I can see the.
Thanks,
pokey
Amanda,
stick
for
better.
the description. Pink blobs are probably mushroom corals. You don't see that in the screen right
That's
now.
the scoop.
We're just
that's
gonna
the
The
scoop.
hockey puck.
Yeah.
After. Is the
Just gonna
camera all the
see.
way out?
Yeah, it is. You've got it in bubble a little bit.
Yeah, that messes me up. But
Well, you're looking straight down.
yeah,
It's not turned.
I'm all fussy. Oh, that was annoying. It's an annoying puck. Oh,
Maybe grab it by the pokey part.
is it what? Grab it by the pokey part?
Yeah. Chris, is the ship still moving?
The ship has stopped.
Thank you.
Oh, does it actually have a...
Set it down and pick it up.
So we're just trying to see how thick these sediments are on this slope. We're about 13 meters from the top of this ridge. Obviously rocky outcrops all over the place here, interspersed with these sedimented areas.
Your pokey stick. Yeah, that's good enough.
Okay. So how long is that stick?
I don't know, Bob. How long is the stick?
Well.
You don't know.
Like.
Oh, let's use the
Wanna
lasers.
Zoom in there, Ed?
Coming in,
So it went all the way to the.
pausing.
Yeah.
So at least 20
10.
centimeters or so.
Yeah.
And it didn't hit bottom, right?
No, I could have.
Coming out.
And it just goes to the white bit there.
Oh,
That's
what?
about 20 centimeters
Yes.
to the white bit. So it's the sediment in these pockets is greater than 20 centimeters deep.
Yeah.
Yep. Okay. So that's.
Good.
Thanks, Bob.
Still full wide.
So it's not just a dusting. Okay, so we're checking for the sediment thickness in these pockets between these rocky outcrops, and it is definitely more than 20 centimeters.
Put it down.
That's all we can tell with the pokey stick. Thank you. Gonna keep poking.
That's the way.
Yeah, maybe.
So did you hit hard ground there?
No.
Hmm.
I need to get a new grab on it.
Oh, okay. Okay, thank you, you guys.
No problem. Nice.
All wrapped up in this.
Okay. So do I just
Okay, moving on.
unstick lock?
Moving on.
Come up?
Let's
Yeah, stick lock off.
All right, come
head to
up.
the mountaintop to see what's on the other side. Good job. Okay, lots of
Bridge nav
slabs.
50210.
They're fairly
So
thick.
Taylor Ann, what's your official title out here? She's the one who's doing all the logging and
Yeah, so I'm the data logger and science manager in training. So this is my 3rd year out with the Nautilus as a data logger. And yeah, it's really great back here just taking all the notes on what we're seeing so that you guys can reference it later.
she's doing a wonderful job
Thank you.
and helping me out immensely. And Megan, who's
I've
been doing a lot of
returned.
the talking. Yeah, tell us about yourself.
Oh, okay. I'm Megan Cook. This is my 9th year of Nautilus with the team. I love it, obviously. I am the Director of Education and Outreach for Ocean Exploration Trust, who is the organization that owns and operates the ship. Whoa, that's a big fish.
Yeah, that's
Yeah.
that's a sablefish, I believe.
Use
It's
them.
We
a
just
lot of
saw
biota.
Whoa.
Pharea sponge as well back there. That was
Oh,
the
Mhm.
cool.
first.
A what kind of sponge?
Pharea
I can't go any more
sponge.
Oh,
direction.
Yeah, I
Sorry.
keep rolling.
Yeah.
we're rapidly approaching the summit here. We got about 5 more meters till we get to waypoint 2. And we'll take a little look around up there, and then we'll head to waypoint 3, where we're hoping to see some bubble streams. The seep site.
Excellent.
Bob, can I get a little push on Argus? Can I zoom?
Yeah.
Thank you. Just a little push and holding.
All right, Susan, folks want to know, how do methane seeps start? Do they develop spontaneously was the question, or are they from a biological or geologic process?
Well, I would say a little bit of both. I
Okay.
mean, probably they're caused by methanotrophins, right, or bacteria that eat the methane, so I guess it would be biological. But there's also there's thermogenic and biogenic
Is my heading okay, Bob?
methanogens, and I think that you should ask
You
Tamara
talking to me?
Baumberger,
Yeah,
our
is everything
chemist,
okay?
this question when she's on
Everything
in the next
too much.
on the next
Other
shift from 12 o'clock to
conversation.
4 o'clock Pacific time.
Yeah.
But generally, I mean, they form because these
Are we still on
bacteria
210?
are
Still on 210.
Okay, full wide
I'm
Argus.
right now I'm at a loss. So
Okay.
I don't remember what the sponge was actually called, but I remember last season we were calling it the Cheeto sponge.
The
I
Cheeto
think.
sponge?
Except that one's
Oh,
not
you gotta
orange,
hold it.
so
Mm.
it's
You're
different.
trying to walk the heading,
Interesting.
so
Oh,
where's that?
it's-
We had
I
passed
slowed
it.
it
It
down
was
so it's not so
barrel shaped, but
snappy,
it had these like
like
little
that was
lobes on
before
the
that I
outside
put
of
in
the
there.
whole entire
Uh-huh.
sponge.
Just gotta hold it a little
Kind of
bit.
looked ruffly.
It's just you're
Like a
moving on a
Cheeto?
set point
Kind
rather
of.
than
I don't know why that got picked up as the common name.
Why does anything get picked up as the official common name? Yeah. So
Yeah.
at a super broad scale, we see these
No,
methane seeps.
you watch the
Because
set
it-
point move, and then it'll
Wow, this is
kind of
looking
let it
interesting.
overshoot, and then it will snap back when you let it off the stick. Yeah.
because of the sediment here, there's a lot for these
You gotta
methanogenic
step ahead, though.
bacteria to process. There's a lot of runoff from the rivers. There's a lot of sediment accumulated in the accretionary wedge, which is the scraped off lead and sediment
streak
from
in
the seafloor subducting.
Oh, yeah.
Here's another one of those sponges.
Oh, wow.
That's really pretty.
Huh.
Beautiful.
I don't know that that's...
Yes, and those bacteria convert all of that to this gas. It can be in a gas phase if it's at less than 500 meters. Actually, you can find these bubble streams in a gas phase all the way down to the base of the slope, which is about 2,000 meters here. But the methane can also be in an ice form called hydrate if the seafloor is deeper than 500 meters. So we're going to be looking for some hydrate to sample with our
Ridge nav.
hydrate sampler, which was developed a couple of years ago at and is property of Tamara Baumberger, our chief scientist.
What?
So we're looking to find the methane in gas form and in a solid form, which is sort of ice-like.
Yeah,
it looks like we have reached, reached the top of this ridge.
Kylie, bring yourself around to 190.
Trying to poke our way around and see what we see. It looks like carbonate slabs, which is also another form that these microbes can live in the sediments, and they can also produce these carbonate slabs. So it's sort of a solid form.
Yeah.
And these are wonderful fisheries habitats. You notice we've seen a lot of biology on our way up this carbonate ridge. So this is also a byproduct of the methanogens, the bacteria.
Yeah, pretty incredible process that you're creating rock as a metabolic chemical byproduct of your reactions.
Right, and that takes a long time. So these ridges are old. They've been around a long time. They obviously don't develop overnight. Whereas these the bubble streams in
As long as you're not an auto
in the gas form are very nefarious.
Oh, okay.
I mean,
That's
they
all that
can
matters.
nebulous. They turn on and off. They move around. The hydrate and the ice phase. Basically, like I said, you'll only see it at 500 meters or deeper. And
it's that time. We're 10 minutes late.
That's something we're keeping our eye on because it has to do with pressure and temperature. And as oceans warm, we think that some of these hydrates will probably be converted from the ice form to a gas form and add more methane to the water column. And methane does acidify the water column. So
this is what the problem is. I got this in front of me instead of this.
And we do have issues with ocean acidification on the West Coast. Of course, and that's one of the reasons we're trying to get a baseline of what's going on out here right now.
Absolutely. The question came in, do methane suits contribute to global warming? And I think that, there are some papers that show that methane released from the seafloor may reach the air-water interface, may dissolve up. But another story is, go ahead.
Bob, can we just stick around here for a minute and just kind of circle around and look?
Yep.
Once we get to the top of the summit here. Yeah, and it's
nav, hold position.
Shallower. We think that the shallower methane seeps probably do breach the air water. I mean, under 100 meters, 200 meters deep. And methane is heats up the atmosphere quicker than CO2.
Yeah.
It doesn't last in the atmosphere as long as CO2, but then it converts to CO2. It can last for hundreds of years in the atmosphere. So, and there is the acidifying factor as well.
Absolutely. So, I mean, part of what investigating these seascapes helps us know is for those seeps that